Okay, so the first part of this chapter is going to be the analysis of single characters in an artificial population. And so these are just some terms that you're going to have to remember and learn. So I've used those a little bit, character, trait, and phenotype. And so it's important you can distinguish between what exactly they mean. So a character is a single visible feature of an organism. So when we describe an organism, when we do genetics, typically we're talking about that there's something that you can see within the organism. So this can be seed color, this could be seed shape, this could be height of a plant. There's something that you can visualize and that can have alternate forms. So a trait is one possible variant of a given character. So yellow seeds would be an example of a trait. The character is the seed color. So notice that a character doesn't specify one of the alternate forms. It just says this is something that can vary. So there's no limit for the number of traits for a given character. So you could have brown seeds, green seeds, blue seeds, etc. In the course of this chapter, though, of course, we're only assuming that there's two variants, and this is just because Mendel, the pea plants that he studied, only happened to have two traits for a given character. And then phenotype is the composite of an organism's observable characters, traits that are under study. So you might be a geneticist, you might have a pea plant, and so you will probably specify that this plant is yellow seeds and white flowers. And so the total phenotype, you're specifying two different traits. You're specifying the yellow seed trait and then the white flower trait. And you could, again, there's no limit to the number that you include, so we could say it's a tall plant with yellow seeds, with wrinkled pods, with white flowers, with terminal flowers, with terminal flowers. And so that whole combination of everything is referred to the global phenotype of the animal. So these are just some of the visual depictions of this. These are the 7 different characters that Mendel studied with his peas. So there is the seed shape, and there was 2 different forms of the seed shape. So the traits were either round or wrinkled. There is the color, so he found that the seeds could have either yellow color or green color. So again, the trait would be yellow and green. The character is just the seed color. Pod shape, so either full or constricted. So this is where the peas, were found within the pod. And so we found that there were some plants that had this full shape and then constricted shape. The pod color could also be different. It could be green or yellow. He also noticed differences in the flowers, so the color could either be violet or white. The flower position could either be axial or terminal. So terminal is when the flowers are at the top of the plant. Axial is when they're not. And then the stem height, they could either be tall or dwarf. So just make sure you remember, am I talking about the character or the trait for any given plant? So how did he make progress in understanding how genetics works? And so it was possible because Mendel was able to create artificial populations for genetic analysis. So he decided who bred with who. And so this is something that we do very commonly in the lab, but where the experimenter determines how an individual mates. I do this in C. elegans, so these are very small worms, 1 millimeter worms, so there's males and hermaphrodites, and I can control which one of these individuals actually mate with each other. Other people do this with mice or fish, and they typically isolate 2 individuals until they have offspring. So we do this because we want to study the genetics and understand inheritance is kind of the power of how we understand genetics. So just some jargon that you need to know. So the first generation is called P or P0, and this stands for the initial parents. And often we will tell you, do the parents breed true? And that means that they are homozygous. So this is an important thing that you're going to find in questions all the time. And so if you ever see parents breed true, you can just assume that they're homozygous. And we'll talk a little bit about what homozygous means. Again, this is review, so I'm assuming that you probably have encountered homozygous before. And then parents differ by 0 or more traits. So typically you will breed 2 parents that might be, for example, you might breed green seed parents with wrinkled seed parents, or you could actually breed the same thing. The first generation of progeny from those two parents are called F1. And then these F1s can also be used as parents, which creates F2. So your population of F1s can be quite large. You can have thousands, for things like plants, for things like C. elegans. You can have hundreds to thousands of F1s that result from a single cross between two parents. And then you can interbreed those together, those F1s together, to create F2s. Or you could, for example, take an F1 and breed it back to one of the parents. So when you're an experimenter, you have the power to do kind of all this stuff. And then if you continue to do additional generations of progeny, those would be called F3, F4, F5, etc. So a monohybrid cross reveals how one trait is transmitted. So this is kind of the basics of how you study how inheritance occurs. And you start with two true breeding parents, or potentially strains. And so again, that means that these true breeding parents are homozygous. But the parents have to be different at one and only one character. So if you look at all of the different phenotypes, they only have one character that they're different between. And so the F1 generation is cross progeny between the two parents, and then the F2 generation is occurred by having the F1 cross to themselves. So this can either be through self-fertilization, which you can do with a plant, and the plant is able to actually produce both the male and female gametes and can produce offspring just from itself. So as an example of this, you could do a monohybrid cross between all 7 of these characters. And so you would cross an individual that has the round P with an individual that has the wrinkled P shape. And so what they found, so we know that they are homozygous, they breed true. So if you self-fertilize the round seeds, they would only produce round seeds. If you self-fertilize the wrinkled seeds, they would only produce wrinkled seeds. And they found that the F1, which is a hybrid between these two, were all round. And then if they intercrossed it, Mendel found that the offspring were both. They found both round and wrinkled seeds. But interestingly, it followed an approximately a 3-to-1 ratio. And so he did this with all 7 of these characters. He found that one of the 2 traits was dominant. So round is dominant, yellow is dominant, full is dominant, green, violet, axial, and tall. All of these F1 offspring all had the exact same phenotype. And in all cases, when he then took it to the F2 generation, he almost always found something that was pretty close to 3-to-1 ratio. So not exactly, this is because of stochasticness and statistics, but it was all pretty close to a 3-to-1 ratio. And so he could explain this because he made the leap to say that each one of these characters was controlled by a unique single gene that had 2 alleles. Each parent was homozygous for one of those 2, and gametes produced by the parents contained one of the 2 alleles, and one was dominant over the other. So let's see how that's done. So here is an example of the tall versus dwarf cross, and so he said that there was a single gene called D that controlled the tall dwarf phenotype. And so because the animals were diploid, each animal inherits 2 different alleles of this D gene. So there is this big D gene allele, so this is also an allele. So it's kind of confusing. A lot of times we call the D gene and then we match the allele name with that. But the idea is that there are 2 different alleles. One we call big D and one we call little d. And so for a plant to be homozygous, it has to have the same allele on both chromosomes. And so there's a big D and a little d. So the first thing he had to predict is what are the gametes that are produced by these individuals. So this individual can only produce one type of gamete. So because the only allele it has is the big D, all of the gametes are going to be the same. And the same thing is true for the little d pea plant, the only type of allele it can pass on is the little d. So again, this comes back to the previous chapter, diploid, and then the purpose of meiosis is to go from diploid to haploid. So the gametes, when you look at the genotype of the gametes, they only have one of the two alleles. So in the F1 generation, the F1s have inherited a big D from the first parent and the little d from the second parent. So 100% of those F1s all have the same genotype. They are all heterozygote with big D, little d. And so this is where the dominance relation comes into play. You'll notice that all of these share the same phenotype as the individuals that have the big D. And so this is what is meant by dominance. So big D is dominant over little d because it has the exact same phenotype as the big D genotype. All right, so now we do this intercross, and now this is where things get a little different, because now it gets a little bit more complicated when we figure out what alleles, what gametes are actually created by this individual. So now this individual has the possibility to create two types of gametes. It can either produce a big D gamete, and that happens 50% of the time, so 1/2 of the time you get a big D. And then it can also pass on this little d, and that also happens 1/2 of the time. So 50% of the gametes that are produced by an individual have a big D, and then 50% of the little d. And then the same thing is true with the other individual because it's an F1 cross, so we're crossing either to itself, but the same logic will hold. And so then if you just randomly pair these, this big D can either be combined, so this big D can either be combined with the big D from the other individual, or it can be combined with the little d from the other individual. So these are the 4 possibilities, and then you'll notice there's 2 ways to make a heterozygote individual. And so the intercross results in a ratio of 1 to 2 to 1. So 1/4 of the animals are homozygous for the big D, 1/2 are heterozygote, and then 1/4 are homozygous for the little d. And so then also if we group them by phenotype, because the big D shares the same phenotype as the big D, little d, we end up with a ratio of 3 to 1. So this is an example of genetic analysis, and this can be used to explain how you get these experimental effects that Mendel saw. So he was able to explain why all the F1 had the same phenotype, why they all shared it with one of the parents, and then he was also able to explain why it was a 3 to 1 ratio. So this also leads to some various terminology on the DNA side. So gene is a unit of inheritance. It's also a sequence of DNA that codes for a molecule that has a function. And then an allele is one of possible variant of a given gene. So here we have seen that there are 2 different alleles of all of these different genes. And in Mendel's rules, there was only 2 different types of alleles. But there's no theoretical limit to the number of alleles for a given gene that can exist. So it's possible to have more than 2 alleles, in the same way it's possible to have more than 2 traits for a character. And then the genotype is the composite of an organism's alleles that influence the phenotype. So if we are talking about a diploid animal, we always have to specify both alleles. And if for some reason we're looking at multiple characters and there are 2 different genes that we're worried about, the genotype is that total composite. So this individual is heterozygote at the A gene, and then it's homozygote at the B gene. So a gamete is a haploid cell that fuses with another gamete to create a new individual. So for humans, this would be a sperm and an oocyte. And then each haploid cell has its own genotype. So it could be A, it could be AB. So this is the case where there's 2 different genes. But what you'll notice is it's haploid, so each gene only has 1 copy. And then genetic analysis is simply the process of determining the genotype-phenotype map by analyzing phenotypes in multiple generations of progeny. So another common type of cross that's done, so we just talked about an intercross. So this was a monohybrid intercross. Another possibility is that you do a testcross. And so a testcross is another example of genetic analysis, and it can be used if you don't know what the genotype is of an individual. So let's say you had a homozygous tall individual. So the relationship of the genotype phenotype was that D was dominant over little d. So if you were a dwarf pea plant, you had little d, little d. So if I gave you, so first just kind of like to make sure we're on the same page, if I gave you a homozygous dwarf plant and I ask you what the genotype was, you should immediately be able to tell me that it's little d, because that's the only way you could have a homozygous dwarf because it's recessive. But if I gave you a homozygous tall pea plant and I asked you what the genotype is, you should already be able to recognize I don't know. There's two possibilities. It can either be big D, little d, or big D, big D. So both of those, because of the nature of dominance, both of those would have the exact same phenotype. And so how do you go about actually determining what the genotype of an individual is? And so the test cross procedure is that you cross this individual to a homozygous recessive individual. So you take this tall pea plant and cross it to a dwarf. And so this, now we have our two possibilities, but they actually would result in different offspring. So the allele that is produced by the homozygous dwarf, this has to be a little d. And if it's a homozygous tall, it has to be a big D. In the other situation, there's two possible gametes that can be produced, big D or little d. And so this would be done with 50% of the time. Again, the homozygous dwarf can only produce the little d because that's the only allele that it has. And so when we do the cross, in one case we have all tall pea plants because it is always going to be big D combined with little d. But in the heterozygous case, 50% of the time you're going to have a big D, little d, and that's when this big D allele is produced. But if the little d is produced, then you would have a little d offspring. And so you'd find that about half of the offspring were tall and half of the offspring were dwarf. So a test cross is a way to determine what the genotype is of an individual that you do not know.